All right, good morning. Oh, that's actually a bit loud, isn't it? It's too loud? Yeah?
Perfect.
So good this way? Okay, I'll try. If it gets too loud or too quiet, raise your hand. Okay, so to start with today, I sent an email yesterday to my mentor students, but actually the information I sent out is probably important for all of you, so I should just go through that very quickly. So, just been told, I know it's coming up, the deadline for registering for VU courses is coming up on the 10th of October, I just heard. Please check, I cannot see it as easily as you can, so check it in VUNet. So that means you need to really start thinking now about which courses you want to follow in the second term. Yeah? In the third term is easy, there you follow biosystems data analysis. But in the second term, you really need to start thinking about it now. For the people who are most interested in bioinformatics, that probably means you will choose the algorithms and sequence analysis course. That's compulsory for the bioinformatics track. Then in the systems biology track, there's basic modeling or basic models in systems biology to choose from. So these are the two compulsory ones. And then we suggest anyone who does the bioinformatics track and does not have a very strong background in programming or computer science to actually follow either a bachelor or a first year chemistry programming course. We usually recommend a Python course, so there's Introduction to Python, which is a bachelor course. And there's also a master course which is called Scientific Programming, but that's a lot less Python, so because we do everything in Python, in the coming things, we slightly recommend the Python course, but the other course is also fine. Yes?
Is it in English?
It didn't used to be, I think this year it is. So I think...
because the name is in Dutch, and then there's a little star next to its name, so I'm not
okay. So in previous years, quite a few people who did not speak Dutch have successfully followed it. I think the problem is the lectures are in Dutch, but the slides are in English. The help is in English, we don't really need lectures, so the book is very good. So what I hear is that actually it's quite easy to follow it, even if you don't speak Dutch. But the scientific programming one is definitely in English. And I thought I actually changed it this year for the Python course also to be in English. But maybe check with the coordinator. Is everyone coming from the other building? Yeah. More or less.
We did a round.
You did a round. Okay, so that's in period 2. Oh, yes, did you move that?
Yeah, I pushed that.
Okay, wait a second. No, that's not. But it doesn't. Yeah. Okay, what I was just saying for everyone who just came in is that you really need to start thinking about courses in the second period. So you need to start signing up for those. It's 2 compulsory courses in the 2 different profiles. Algorithms and sequence analysis for bioinformatics, basic models and systems biology for systems biology, and then typically you have 1 additional course. You can also do both track courses. And then apart from that, there's a couple of other options that are possible to follow. You can always go and shop yourself, so you're free to look in any other master course for suitable courses. And the only thing I could find was quite technical in computer science, so it's not too applied. So if you have a sort of computer science background, something like software architecture may be interesting, but maybe a little bit too much if you have no programming background, so then you should definitely start with a more introductory course. On the biology side, there's also a course called Signal Transduction in Health and Disease, which is very nice, and the material is nice, but you have to memorize quite a lot. So people who struggle with memory of a lot of facts, and maybe that's not the course for you. All right, is there any questions on that? Yes?
Do the ECs for the lower level courses also count for your masters?
We have the rule that you can have one bachelor course in your master, just because we know you come from such diverse backgrounds. So you're allowed to have one bachelor course counting towards your master's degree. Yeah.
But if you'd like to do both compulsory courses for system biology and bioinformatics, and also do the introduction, is that possible?
I think that's quite heavy. So to do 3 courses at the same time. Both basic models and algorithms of sequence analysis are quite demanding courses, although theoretically. I think introduction to programming is a lot of work. So I am not aware if anyone in the previous years have successfully followed all 3. I don't actually think so.
But the introduction to programming, it takes, if you don't have any programming experience and you're slow on getting it takes 18 hours a week. Because
Yes.
you really have to do a lot of practical
So
work.
indeed, all 3 think on the order of 50 to 60 hours a week workload. Yeah. So if you also have a side job or something, then that's definitely possible. Also, I think there may be quite a lot of overlap with compulsory classes.
Yeah, that's also
Yeah. So it's I think it's a tough one. You could try, but I think it's tough.
But the other option is that course material freely available to us, so that we can just learn more afterwards, because we're going to use more Python. We can fall back on it.
In that case, I would strongly recommend just following a Coursera Python course, which is kind of meant for distance learning, which is fine. There's also a good Khan Academy Python course. So in that case, I would actually follow one of the courses that is meant to be followed offline. Or
what was the name of the second one?
I think it's the Khan Academy. People are quite enthusiastic about. Anyone done that? People are quite enthusiastic about those programming courses as well.
Yeah, they have everything. Having history, geography, morgen. Hoe gaat het?
Ja.
Heb je lekker geslapen? Heb je lekker geslapen?
Alright, so if you're not sure about your courses for the second period, please do go and discuss with your mentor. In the VU system, it's not so bad to sign up for 3 courses. So if you're really not sure, you can start with 3 courses and then drop one of them while you're going on. So that's also a possibility of doing it that way. I think there's a strong possibility that introduction to programming in Python is going to be filled soon. So if you want to do that, I would suggest you register for it quite soon. And then another thing to consider is that in period 4, 5, and 6 at UVA, you have to register for all of these 6 weeks before the next semester starts. So that will be, I think, half November. So if you, in the second half of the year, want to follow any courses at the UVA, you also need to know much sooner than you may be thinking. So towards the end of this course, you should already start thinking about the rest of your year, so you can sign up in time for the UVA courses. Yeah. Okay, so let's start with this lecture. So today we're going to look at protein structures, which you've probably already seen a bit on the fly in the previous lecture, and to profiles. So actually what we're going to do is maybe zoom in a little bit on some of the concepts you already seen coming by quite fast. Maybe just to start, why do you think we have this lecture in here? So why do you think, in terms of the project that you are doing at the moment, so benchmarking BLAST against PsyBLAST, why is it important to think about structures and profiles? Anyone, any idea? Talking about, yeah?
To get an idea of what you're actually looking at.
Yes.
Of what you're actually working with.
What is the relation with structure to the project? Is there a direct relation? Yeah.
Structure of proteins work with function. So if you work with GO, I mean, and those function.
So you say there's a strong.
Structure, but you with scope or PFAM, there's some structural information there as well.
Okay, so there's, you say for Go, there's a strong relation between structure and function. So you should know a little bit about the structure. Scope, does that contain any structure information?
Yeah.
A lot of it, right? And actually it's really based on structural information. And I actually show you why that's actually such an important tool later in the lecture. And then PFAM, is that based on structures?
Yeah, the main.
It's not really actually. So it's really yes, exactly. So it's really sequence based. But there's a small caveat there. So how PFAM is built, and you've probably already discussed that in the last lecture. If you generate these sequence profiles of domains, and this is also based on sequence searches, but there's a very big but. For a lot of the PFAM families, the seed alignments, so that's the alignments to actually feed the further search, they've actually been hand tuned. So someone's literally gone through them by hand and to check if all the residues in the profile are on the correct positions. Very often they would use structures to actually curate these alignments. So to some extent, structures may still be involved. Alright, but the first thing I want to do with you, yeah?
I don't think you can see the recording.
No, but does that mean that you also don't see it on the lecture later on?
Yeah. Watching it, so it's easy.
That's really bizarre, so I think. It's really annoying that it's on the screen. I don't know if I can change that around. Do you rather want only the screen? Because I can also record it in such a way that I only have this part, but then you don't see the slides as well. So I think it's usually more helpful to see the slides to get rid of the voice.
Yes.
Yeah? Okay.
Sorry.
Okay, so then we'll just do it like this. It's just annoying you see me moving on the side. Now. Yes. Alright. So BLAST. And actually typical BLAST output. So you've all seen this type of BLAST output by now, right? So you have a bit of a feeling what this means. What's going on here in this region? Gap, yeah? So I've got a pointer here. So this is your query, right? Here there's no gap. So what would that mean? What would be there? What's happened in evolution? An insertion or deletion depends on which. So here, with respect to, so here is part inserted with respect to these sequences or vice versa, yes? Do you think at any point in a sequence you can just like start adding or deleting things in your, no? What would that depend upon?
Functional region or not?
Functional region or not, yes? Anything else? Yeah, that's where you have higher, sometimes in exonic boundaries, for example, you get more easily insertions and deletions, but that's more an evolutionary point, not so much from the protein perspective, yeah?
Repetitive sequence or bounding sequence?
Yeah. Okay, we'll maybe come back to that one at the end. Because there's actually, so what you see is often that in regions where you get deletions, like here you see many more deletions, even if you see in between sequences. So for example, here you see a deletion here, but not in the other positions. This is quite common. So the thing is, the same region, apparently you're allowed to take a part of the sequence in or put it back out. And what you also very often see is that the length of the gap is a little bit different. See? So there's some variation in gap length. So apparently it doesn't have to be exactly this, but you could have a bit of variation there. Alright, so if you look at this type of output, and this is actually done after a multiple sequence alignment, which is actually a method to make these alignments even better, which you'll see all in algorithms and sequence analysis. You see that there's some residues that are extremely conserved, right? So you see this K here, it's actually there in every single sequence. See that? And we have a couple more here at the very, here's another. Okay, that's not completely, you have just the same charge residue here, and R arginine. Also, so you see a couple of, and here and R, but also very conserved. Yeah? So you see a couple of these positions are very conserved through all the protein families. And now this is actually one of the most important ideas if you want to make sequence searches more sensitive. Because what if I'm now going to find a new sequence and it doesn't have a K here and it doesn't have a K here? Do you then believe that that belongs to the same family or not? Or may it just be that some of the other residues have matched by chance? Well, if these are functional or structural, very likely they will not actually belong to the same family. And so actually putting extra weight on residues that align in all the other sequences really helps to make matching alignment scores to some extent more sensitive. So to that end, and we already saw this in PsyBlast at the end of the BLAST lecture, we have something like a specific position-specific scoring matrix. And so this is a matrix where you have the amino acids on one side and the sequence positions on the other side. And so you generate a PSSM on the BLAST output, and then you can use this PSSM with a new search with BLAST to search against the database. And so in this case, you don't align the sequence anymore to the database. So that's a very important difference between the second iteration of PsyBlast and the first iteration of BLAST or PsyBlast. It's not the query sequence you align against the database, it's actually the PSSM itself you align against the database at each position. Yeah?
I think I also read that the PSSM replaces the substitution matrix.
Yes, exactly. So it basically replaces the sequence and the substitution matrix. Yes, that's exactly it. Now, what is done though is that the substitution matrix is not completely out. So if you have very little sequences, actually the substitution matrix is used to initialize the weights in case there's very little information on specific positions or because you found only a few sequences. Otherwise, if you just find a signal sequence, you're just going to put all your weight on that sequence, and of course that's not what you want. So it's weighted with the original substitution matrix score. But effectively, yeah, the substitution matrix as was is not used anymore. Okay. Some questions here. So say we have a protein sequence of 100 amino acids, and we run BLAST. We obtain 25 hits to the sequences, and these hits range from 20 to 250 amino acids. And from this, all we build a PSSM. What are now the dimensions of the PSSM? So what do we have on one hand? How many matrices and how many on the other side? Yes, there is a bit too much information here, but that's yeah.
I would say it's 100 on the x-axis.
Yeah, so it's 100 on one, and how much on the other side? 20. So why 100? So what do we do with this longest, that 250 length sequence that's also found, so that it had a hit? What's happening to the remaining amino acids? What do we use for the PSSM? Because that doesn't fit, right, in that 100.
Some sort of gap or insertion score?
It's actually simpler than that.
It's not aligned.
It's not aligned. So you don't actually only use the aligned residues of the hits to build a PSSM. So this is also a real difference between something like a multiple sequence alignment, where you really try to put every single residue against every other residue. Whereas BLAST, when you get BLAST output, like the first one, this a query anchored alignment. So actually, the only part of the sequence we care about is that what's actually available in query. So if there is a gap here, like for example you see this split line, it means there's actually an insertion here we won't take that into account to build the PSSM. Yeah.
Why should the y-axis not be 20? Because I understood.
So there's 20 different amino acids.
There's 20 different amino acids.
Okay, but there's 25 hits. So
Ah, oh, but you, yeah, but so that's just, so the amount of hits just go into the statistics. So you don't build a PSSM for every single hit you've made, yeah?
So, sorry, so
Yeah.
the 25 hits, sorry, there are 20 amino acids.
Yeah.
That makes the matrix, and then how do you get 200? Because that part.
To 100. The sequence itself, the query sequence is 100 amino acids. How do you see? I thought I had a. So here, for example, so on the one hand you would have your full sequence, on the other hand you would just have listed all the 20 amino acids. But it's your full query sequence that's listed, and then the statistics of the aligned residues are taken into account, and not the insertions that don't actually align to your query. That's also different if you go to even more advanced methods. Something slightly different happens there. And for example, you can also start looking at gap positions, and you can imagine that becomes even more sensitive. The PSSM can't do that. It only looks at the weights of the sequence or the amino acids observed, and not, for example, if there is a position, like here for example, where a lot of insertions or deletions have taken place, because of similarly as to a place here where we say, well, this K is extremely conserved here, we actually also see that these gaps reoccur in the same positions. We can actually imagine that you would also have that help to build your subsequent alignments and be more sensitive. So it would be really nice if on these positions here, it actually could lower your gap penalties. Now that's not exactly done here. That's something that is being done in hidden Markov models and HMMs. We're not going into that today, but for PFAM, of course, they are constructed with that, and that's also why they're more sensitive. Okay. This one, question 2. So if we would start the blast run instead using the sequence of one of the 25 hits, would we then get an identical PSSM or not? Because we're looking for the same protein family, right? So we hope to get the same. No? Say? So what could, for example, how could it be different?
The length of one of the hits is
Yeah.
different, then you get a bit.
Exactly, so if you can already see, right, so if you take the 250 one, you'll get already a completely different dimension. It may also be that you actually get, if you run that blast hit, you'll see that also, it's not, so if hit A finds hit B, it doesn't necessarily mean that hit B or sequence B also finds sequence A. Yeah, so you can actually get different things here. Alright, so this is what the PsyBlast iteration looks like, right? So we run the query against the database. We have obtained hits here. Then very important, we set a threshold. Then we build a PSSM on these significant hits, and maybe you should actually add a little thing there, so not only significant hits, but the aligned residues of the significant hits. And then with the PSSM, and not with the query sequence, we run blast again against the database. Yeah. And then you can iterate this quite a few times. So why is this threshold so important? Yeah, so what you can get something is called profile wonder. So if you start including false hits, you may actually start including sequence signatures of a completely different sequence family, right? So if you set this threshold in PsyBlast really lenient, and maybe you would go up to something extreme like 10 or something, you will get actually sequences in there that are not correct, and so your PSSM really can turn off. And what you can actually happen then, because there are some sequence families in databases that are extremely large. So some sequence families only have maybe 100, 200, 300 examples in the large databases you're looking through. But there's usually some protein families that maybe even contain 20% of all these sequences available. So one of these examples is skin barrels. They're a special type of enzymes, and a very large part of all protein structures actually, and also all sequence families, looks like that. So if you just get one of those, your profile just may wander into that direction and start just collecting all these skin barrels. Yeah, so that's why you have to be very careful with this threshold here. Yeah? You may, so what you don't have to. I think you have to rerun manually blast every time. So if you want to, you could experiment with that in a project. Which way would you go? Would you slightly increase it or lower it? Making it more lenient or making it more strict towards the end? More strict towards the end. I would probably do the opposite. So I would actually maybe then have a last run that was less strict, so that it can't make your profile or your PSSM in some ways dirty anymore, but you may still be in, so that you get completely in the wrong direction, but you may still see, there may still be some significant hits in this relatively high E values. So you could think about the last run of your PSI-BLAST threshold. And actually, if you look at PSI-BLAST carefully, you see there's two thresholds. There's one for reporting, and there's one for inclusion in the PSSM. Have you already seen that? Question?
Yeah. So would PSI-BLAST stop after so many iterations? Say you have it on at 10, and after 5 iterations.
So say you put it to a 50, so go a bit extreme.
Yeah.
Is there a natural point that it doesn't make sense anymore to run PSI-BLAST?
Yeah.
When? So if you run it once, you have your PSSM, you put a PSSM in, and you get exactly the same list of hits above your threshold, right? You're going to construct exactly the same PSSM, so you can keep iterating, but you're going to do exactly the same. It's not going to change anymore. So actually, PSI-BLAST does stop when that's happened. So it tracks if it finds any new hits above your threshold. So even if you put it to 20 or 30 iterations, it probably will most of the time not make that amount, simply because it will not find any new ones. Yeah. Right, so this is this idea, right, of false positive. So if you have like a false positive already in your profile, you may not end up with a very nice profile in the end. Okay, this is a very typical scenario we'll see in not algorithms and sequence analysis, but of course after that, structural bioinformatics, where we look at a lot of protein bioinformatics. And so what we would typically do there, and we will do that many times, that you have a protein sequence without a known protein structure. And what you will typically do in such a case is that you will find a known structure by blasting it against the PDB, which contains about 70,000 proteins at the moment. But we run blast against the PDB, and we found 0 hits. This is very common because there's unfortunately still many proteins for which we don't have a good example structure in the protein structure database. Yeah? So that was blast, and so you have this. Now can PSI-BLAST help in this scenario? No, so who says no? So why not? Yes, so if you found 0 hits, we have nothing to build a PSSM. Nevertheless, we can use PSI-BLAST. Who's got a really clever way how we can use PSI-BLAST here?
Maybe if you up the E value for the inclusion threshold for the PSSM, maybe you can then get some hits because you're a bit more lenient.
So we know that the first hit has an E value of 100 and it's terrible, so no, that's not going to work. So you could do it, what you can do is indeed you can go through manual inspection and see, like, well, maybe we can find some evolutionary clues, for example, we know, for example, already functional residues lie because we have the protein structure of the template and we can see if those match. I mean, you could do that, so you could actually look at the alignment by hand and still consider a hit that is, despite having a high E value, you can still include. There's something much more clever you could do.
Could you make a PSSM
with your own protein structure as well. So you duplicate your protein structure.
We don't have a protein structure, so we have a sequence for which we don't know the structure and we want to find the structure within.
Try a different database, or do we only have this database?
Do you only have this database, or?
Oh, right.
So what database? Anyone? Yes.
You can generate your sequence and then try to find matches from those sequences that you generate yourself. It might look different since you don't have the structure, but you have the amino acids that are similar.
I think we're more want to go that way. So different database. So what type of different database? Anyone? Swissprot, Uniprot? Yeah?
Because it's an idea to use a sequence to put it in the program that it can predict the theoretical structure of the program.
Yeah, but they actually work with things like Cyblast. So they actually have this kind of idea inside.
You have to use cat or Scott because they look at structure.
Because they only have structure, so they only contain a subset of the 70,000 structures in the PDB. But I heard kind of the right answer here, so someone suggested, so why don't we just take all the sequence genome? So at the moment there's millions, so this is 70,000 proteins. We have millions, I think on the order of hundreds of millions protein sequences available at the moment. We just don't know the structure for all of them. We can actually generate, so in the first round we can use several rounds where we have the first database to be Swissprot, Uniprot, which is already a sequence database. You don't necessarily have to have a structure to be a Uniprot. And then you start generating a PSSM, so you make a really sensitive PSSM, and we will do 5 rounds of this. And then you just take the PSSM, you take it out in the 5th round, and then you run it over the PDB. And now suddenly you may find hits, because now you have a very sensitive PSSM that can go much further than BLAST could do by itself.
Sorry, I forgot to. The first step was to run it in a bigger database, right?
You first do 5 iterations, say, on a much larger database, on which you can actually. So that means you'll actually find hits, right? So there will be. I mean, there's hardly any proteins. It's very unlikely. So unless you get extremely rare species, we have a lot of sequence bacteria. Eukaryotes. It, so the chance that you have find a related protein sequence is high in these large databases we have. It's just that for many of them we don't have a structure yet, or at least not a structure that is similar. But if we then construct a PSSM on this very large database, we have a very good evolutionary profile. And then we can start using this trick of knowing where exactly the most conserved residues are and run that on the PDB. And then you can see if that fits well. So can Cyblast help? Yes, but not directly. Yeah. This is also, by the way, a trick you could actually do on your project. A lot of groups don't get to there, but it's a nice thing to try. Okay, so which database should we run Cyblast on to create a PSSM? So how would you answer that now in an exam? Is there a single answer here? No. Yes, there is. Sorry? The largest you can get, yes. There's only one exception to that, or you're not allowed to have identical copies of a protein in it, so you have a non-redundant database. So, and that's actually also what's prepared typically for Cyblast. So if you download Cyblast, then that's what you'll get with it in a database. It's the NR database, non-redundant database, and it's one of the largest sequence databases available for proteins. But the exact sequences that have exact matches have been filtered out, so it's been made non-redundant. So there's now 2 identical sequences in there. Yeah, because if you have 2 identical sequences, and especially if you're thinking about these timbral families, you're really going to dominate your PSSM with that. Okay, in this one we actually already discussed what E-value threshold should be set for inclusion in the PSSM, and typically this should be relatively conservative. Yeah.
if you had such a big database with a lot of sequences, wouldn't you also assign conserved parts that are not actually conserved, that are just that many different sequences?
So the idea there is that you use a strict e-value to start with to prevent spurious hits. Yeah. So you use a large database and a low, so a very tight e-value cutoff. You're very strict in this case. Yeah. All right. You already looked at some benchmarking papers yesterday, is that right? You did the benchmarking lectures. Did you see this figure, roughly? Yes, this is inside your paper, right? Okay, so what you see here is that you have BLAST. It doesn't do particularly well in this graph. Then you see PsyBLAST, the magenta curve here really does considerably better, right? And then you see a lot of them also outperform, but there's a huge gap here. So this PSSM idea really helps. It really makes BLAST more sensitive, and a lot more so. Again, I'll show you in the end why this is. And here is so the profile, and at the moment we're even a little bit higher up because now we have profile methods that are even a bit more sensitive. So you align a profile on the one hand against the profiles for all the database sequences. There are also profiles made, and so you end up aligning profiles against profiles instead of profiles against the sequence database. And if you go to these type of methods, you now can even get a bit higher. The real trick of those profiles is that you also include the gap probabilities. So you basically you can specify the probabilities of insertion and deletion on specific positions in your sequence, and that's something that the HMM can capture. That's why you can get so much more. So here we already actually looked at, we considered this, so a sequence profile can help to find distant homologs, and why does it work? And we have considered this, so but you get these extremely conserved residues, for example a G and a D and a lysine here. Now some of them are functional, but the majority of the very conserved residues are not necessarily functional. What's actually, why can't they be changed?
Functional, they're structural.
So they're structural in here. So what, for example, could be going on with the lysine on an important place? Hydrophobic one, we'll look at it later. So it could be that the entire protein doesn't form structure anymore. If you maybe have so the most clear one, which is the bachelor students already saw that, you have a cysteine, you have two cysteines, you have a cysteine bridge, so cysteine makes a diversified bridge with another cysteine, and these are extremely conserved because if you take the cysteine bridge out, the protein structure collapse. So a lot of the very strong conservation you see in these sequence profiles is actually based on the structure. So a lot of the evolutionary history you see on conservation is just the ability to remain folded of the protein. And that's also why protein structure is so important. Okay, let's go a little bit further. So we'll go to some of the basic structure in a minute. But to start, we have the conserved residues, and here you have an alignment, and you can see here actually in detail what happens. So here you have a much smaller alignment. You see a subpart actually of an alignment. You see here a loop, and you see, for example, this proline here that interacts with some other residues is extremely conserved. And this E here is as well. So it's E here. Yeah, and this proline is sitting here. Okay, so the next part is probably a bit of a repetition, so I'll actually go to So for who is this all completely trivial? Like, can you do hands up if this is like primary, secondary, tertiary, quaternary structure? Yeah, exactly. That's over half of this room, I think. So some of this is going to be a bit of a repetition for you. What I would really like you to do is think very hard about, because we'll do that later, what types of computational problems we would need to perform. So there's a lot of computations on all these levels of protein structures. What kind of computational problems maybe need to solve or can be solved only using a computer. So we'll come back to that at the end of this section, but people just raise their hands, try and think of some things of like what you can algorithmically do with these structures. Okay, and then also think like how would you actually solve it? So if you were to do it, would you use something like alignment, or would you just be able to do it by hand, or how would that work? Okay, so the levels of protein structure. So this is something, this kind of very basic structure, this is still kind of exam material. So we don't say a lot of depth in the biology necessarily you need, but this is something you would need to know to be able to reason also about things like blast, psi-blast in relation to FoB or BFA to answer questions in the exam. So this is basic knowledge. So if you're very unfamiliar with this, it may be worth reading up a little bit about it as well. So the primary structure is just a sequence. So just the sequence of the 20 amino acids. So that's just what we see all the time when you have a FASTA file, for example, right? That contains your primary structure. Now the second level of structure is secondary structure. We'll look at that in more detail in a bit. But what you see is that, for example, where the sequence starts to curl up in a regular structure. It's not actually necessarily the amino acids, so the sequence of the letters that generates the structure, it's the backbone, so the parts that's the same between all the amino acids that allows the structure to be there. And I'll show you that in a minute as well. Then the tertiary structure is the fold, so that's how these secondary structure elements here in blue arrows and in these red cylinders are folded together. So this is really the fold. And then the quaternary structure, you will have protein chains. So you see here two different protein chains, two different proteins. So that will be typically, not necessarily, residing from two different genes, and they have in two different proteins, and they can also start interacting together. And many proteins in our body will actually interact in very large complexes to be functional. You'll also have proteins that function with different partners at different occasions. So that bind to a different protein, for example, the phosphorylase part of the protein and has a different binding partner all the time. Yeah? But typically it needs interaction with other proteins, other molecules. Think about the transcription factor binding to the DNA to regulate gene expression, or an enzyme that binds to a small molecule to metabolize a reaction. Okay, so the first part of the primary structure of the protein, that's more or less a solved problem, right? So if we have sequenced the DNA from a species, we know where the reading frame starts. It's not always quite as easy. And we can basically just use the codon table to translate that into amino acids, right? So if you already done the programming exercise on the codon table, we're going to do that. But this is something that you'll not find it when you program it yourself, may not be quite as easy, but this is something we can at least by hand think of. This is quite easy. So once you have the sequence, we can translate the DNA sequence into proteins, or into a protein primary sequence. So scientifically, this is more or less a solved problem. All right. So then the primary structure looks something like this. So you have a backbone. Here the backbone is highlighted. And here in small you see the different residues, so the different amino acid types. And you see the chemical composition for all, but two actually, of the amino acids is exactly the same. So you should just really see this as a chain with 20 different colored beads, which like different properties liking each other in, or actually repelling each other in some cases as well, with a very regular backbone in the middle. And if you look at it more detail, so here you have the backbone in pink or magenta, and in the blue you see the chemical structures. So no need to know these by heart for the exam, with possibly 2 exceptions. But what you do need to know is the group. So you typically have a very large group of hydrophobic amino acids. Then you have the charged amino acids and the polar amino acids. And basically, protein folding, we'll say later, is dominated by the difference in these types of amino acids. So it's really the types that make the difference. You also see this very irregular backbone structure again here. And the R is here is where the blue parts of the rest groups are here. Okay, so this is what you roughly need to know. There's 3 special types of amino acids. Cysteine I already briefly mentioned. So it can actually make an additional bond. So here you have the backbone, here you have a cysteine, and here you have a cysteine. It actually makes an additional bond, a covalent bond, through the structure. So you can imagine this really helps to stabilize the structure. It's typically only extracellular though, so not all proteins can contain these structures. Then you have proline. Now proline actually, other than the other residues, where's the proline? This is it. Sometimes it's with the charged. Oh, is it not? Oh, here, yes, exactly. So here you see, it actually folds back. The chain where the other ones are falling straight out, this one actually folds back onto the backbone. You see that? So that means that actually the backbone itself is a bit more restricted in the conformations it can take. And glycine is the opposite. Not only it has no real side chain, so it just has a H sticking out. So it's very small. And that means a similar thing, but that means it's actually way more flexible at the position than the other amino acids. So protein and glycines can actually really structurally change the property of the backbone. So for example, if you have a helix, we'll see that later, often there's a proline at the top to stop it because it disrupts the helix. Alright, and then maybe the most important part in protein structures is our hydrogen bonds, which come back at several different levels. So hydrogen bonds, probably have seen this in high school chemistry, but it's between a delta negative group and a delta positive H. So you have typically an N or even an O, so this is also a delta negative group. And then between a delta positive H, you can form a very weak transient bond called a hydrogen bond. So it's not really like a covalent bond, but it is an interaction that's relatively energetically powerful. So you wouldn't have these broken in real life systems. This is, for example, so water. I think I have the next one. Water contains a lot of hydrogen bonds. So actually, the reason that water, which is actually very light in terms of its atoms, is actually liquid at room temperature, is actually just because these hydrogen bonds are quite strong. So many similar weight gases, like methane, for example, and ethane, they're gas at room temperature. Because these hydrogen bonds are so strong, it actually is kept together as a liquid at room temperature. So hydrogen bonds are strong. Alright, so also in the peptide backbone, there's these delta negative atoms, the O's in this case, and there's delta positive H's here, the NH group here. So this H is delta positive. And these hydrogen bonds want to be formed. They basically always want to be formed to some kind of extent. Yes?
Are all the double bonded oxygen atoms, are they all delta negative?
Yeah. So this is something that, so here is there only 2. This delta negative O and this delta positive H is in every amino acid of your backbone. Yeah? So all of these want to form hydrogen bonds. Now one of the easiest ways to start forming these hydrogen bonds, and this was actually already thought of even before, because we knew the chemical structure of proteins or amino acids before protein structure was experimentally seen and observed. We actually already realized that you could generate, within the constraints of the other atoms, quite nice regular structures, specifically a helix, like this. You can have like a spiraling form in which you can satisfy then all the hydrogen bonds of the backbone. In fact, before the first protein structure was thought, they thought that proteins were going to be a really nice regular structure, just like DNA. But when they then saw kind of the helices, but also these other loops and things, they were actually quite disappointed that it wasn't such a nice regular structure like DNA. It's probably good though, because that's probably also why proteins can perform so many different functions, such specific shapes. So this is the helix, and our data one here, and probably you see actually the hydrogen bonding between the H and the O group there. That's very regular, and they're all satisfied. Now another way of satisfying is by beta strands, so you can do it locally, like in these helices, but you can also in beta strands here, you can actually go across two stretches of the backbone and you can form hydrogen bonds like this, between the acceptors and the donors. You can do this in two ways, so you can have the change going into a parallel or an anti-parallel fashion. So you have two types of beta strands, parallel and anti-parallel. And now the last part is the loops. And loops tend to be much more flexible, but they still have hydrogen bonds. So how can they be satisfied? So they still have these donors and acceptors, right? And so we want to also generate hydrogen bonds for those atoms. And so how's that going to happen? Yeah, it says it actually. Yeah. So, but can you imagine it? So it's the solvent. So the solvent can have hydrogen donors. The backbone. So you may still get to another position in the backbone, but just on just a single one. But the side chains. So if we go back, most of the polar and also some of the charged ones can actually also have donors and acceptors in there. So it can actually also be side chains that satisfy these hydrogen bonds in the loops. Alright. And as I mentioned, there is also hydrogen bonds in water. And this is exactly why we have liquid water. Now it's actually the hydrogen bonds in the water that make the protein fold. So it's the water and not so much the hydrogen bonds in the backbone that really make that this protein curls up in a kind of three-dimensional structure. So the most important effect, and we also know this now physically, some debate over it for a while, but we can now kind of measure this also in quantitative sense, is that the hydrophobic effect. So this is just the effect that oil and water don't mix well. So you put oil and water in a cup and you shake it, and eventually you'll have the oil floating at the top again, right? So the oil, which are C chains, and those don't contain, so this is a very long carbonyl chains. Here there's H's, but these are not positive, so these are not hydrogen bond donors. And so these cannot form hydrogen bonds with the water. Yeah? And so what's happening is that the water prefers to be more around other water molecules than to be around these oil-like molecules. So another way of saying a hydrophobic effect, so what's happening is that the interface between the water and the oil tries to be minimized. Yeah, so in this cup, that's just a straight line, right? So now what can a protein with many hydrophobic chains on there do to minimize its surface with the water? It goes compact, right? Collapses. And then that's exactly what happens. So you get this kind of hydrophobic collapse. Or if you have just two molecules first that are hydrophobic, they would clump together. And so in a protein, if this is a very simple picture of a protein in the black layer are the hydrophobics, they will curl up in the middle of the protein. And if you look at protein structures, if you start visualizing, this is exactly what happens. If you were to just color the hydrophobic residues of a protein, and this is exactly what I've done here, you see that they're really nicely centered and make a very nice tight packing of these hydrophobic groups in the center of the protein. So this is the main driving force of actually the vast majority of proteins. And you can imagine that if you suddenly put a K or like a strong charged amino acids in here, that's going to be a disruptive change, and that protein isn't actually going to fold anymore, so it won't take that structure anymore. Right. So then we have the tertiary structure, and that's basically together with the secondary structure elements and the hydrophobic effect to come to these kind of structures. And things like SCOP at the topology level, the fold level, is really what describes this. Yes? Yeah, I need 2 more slides. We started a bit late, so that's why. I think it's going to be the next slide. Yes, it is. I'm just going to get to there. Okay. So why do proteins actually fold? So it's oil in water, but there's also a little bit of secondary structure. Yeah. All right. So I do have one question for you before the break. So why does secondary structure actually form? Because if you think about it, the hydrogen bonds in the backbone may as well form hydrogen bonds with the solvent. And now there's a little bit of data over which is stronger, and the hydrogen bonds in the secondary structure may just be slightly stronger than the ones in the water, but that's not the main effect. There's a different reason why you actually get the hydrogen bonds or the secondary structure forming. So think about that for a minute. We'll have a 10-minute break. We'll start again at 10 past. And think about that question. So why does the secondary structure actually fold if the hydrogen bonds aren't that favorable compared to the water?